Hello and welcome, dear students. How are you all? So I welcome you to the class of biology. Good evening, everyone. So centers, please report your attendance. Itanagar, Shillong and St. Paul's. I would like all of you students to please report your attendance so that we may get started. Hello, Shillong students. How are you? Hello to all. All right. Yeah, okay, Shillong 1, 2. I can see 2 students right now. So I believe more are coming. Right, yeah. Other centers also, please report your attendance. And we are going to begin now. So in the last class, I told you that I will clear your concepts in the next class about the transcription and translation process. Okay, only one. All right. Okay, I believe there was somebody else also. All right. Okay, so in the last class, I told you that I will clear your concepts regarding the transcription translation process, and there were a few doubts also. So I have some PPTs to help you out. I had made some PPTs. So let's begin with them. So regarding transcription and translation, this process takes place in a very systematic manner. So we are going to see here how DNA is making RNA and how RNA is making protein. DNA synthesis, DNA to DNA synthesis, we have already done many times. So right now what we are going to do is just DNA to RNA, and from DNA making RNA, then making protein. So we all have a basic understanding of this fact now, that the genes are there present in their DNA, and they have the information to make the proteins. So basically, it's the information contained in the DNA that we have going to that we are going to exploit, that we are going to use. So the cell first of all makes messenger RNA, and these messenger RNA are copies of the DNA genes that are needed. Messenger RNA is read at the ribosomes, and these ribosomes are present in the cell, or they are present in rough endoplasmic reticulum. This is how proteins are produced. This is a brief summary. Okay, so let's look at the details now. We need to understand a few things here. What is messenger RNA? Messenger RNA is carrying the information, and it looks like this in this figure that we have here. Ribosomes, they have certain things and some structures that are represented in this diagram. So ribosome has a small subunit and it has a large subunit. The large subunit has two sites, two places where two amino acids can sit together and they can form a bond, right? The large subunit has a site where two people can meet and they can form a bond together of a friendship or something. So transfer RNA amino acid binding sites are present here, labeled 1 and 2. The small subunit is going to be the place where messenger RNA is going to be read, right? And the ribosome large subunit also contains a catalytic site that is being represented as a top corner in the diagram, right? Okay, so this is sufficient for you. Right, let's move on. So how does transcription begin? Transcription begins from one strand of the DNA, which is known as the anti-coding or non-coding strand. The coding strand does not take part in DNA transcription, right? So here we have a blue colored structure. I am calling it RNA polymerase. So it binds to a promoter region of DNA near the beginning of a gene and separated near the double helix near the promoter, right? So this is the enzyme responsible for making messenger RNA copies of genes, and DNA is going to unzip. It is going to separate the two helices together from each other, and that is at the site of gene that is needed. Transcription. Second step. First step is already initiation. Initiation is the binding. Second step is reading and moving forward. So this is the elongation step. So the RNA polymerase is making RNA from only one strand of DNA. See in this figure, compare this with this previous figure. In this previous figure, we have just started, and in the second figure, you can see we are making RNA from only one strand. the other strand is not being used. You can see on the upper strand there is no double stranded structure there, right? So only one is being used, right? So only one strand is being used. You can see the red colored structure, that is the messenger RNA, the red colored filament like structure. Everybody can see that, I believe, right? So you can see that it is very clearly visible here. So transcription, compare this with the first step, initiation, the RNA polymerase binding to the DNA. The RNA polymerase binds to the DNA and then the chain elongation will start. Only one strand will be used. What is the name of this strand? It is called a non-coding strand. The coding strand will not be touched at all. You can see here in this figure it is clearly visible. Alright. So the template strand or the one strand will be used. Okay. As we move forward, there is a terminator site, there is a promoter site, there is a termination site. So the termination signal terminates the process of formation of RNA from DNA. Okay. So as soon as it encounters this, with the help of a release factor, the RNA polymerase detaches from the DNA and releases the RNA molecule. The details of this I have already given you in my previous class, right? Okay. So conclusion, after the transcription has happened, the DNA will zip together. Previous step was unzipping, now it will zip together. Both the strands will come forward and they will form the zip line or the barrier, or they will form the bonds together, hydrogen bonds. And the RNA polymerase, this is the big structure on the right hand upper corner, it has separated. And the messenger RNA, in this slide I have written only RNA, it's basically messenger RNA. So the messenger RNA has separated, right? Messenger RNA has separated and it has this kind of structure. Okay. So the RNA molecule is now free. This RNA molecule was synthesized inside the nucleus and now it will move out from the nucleus through the nuclear pore. Okay. Now let's consider translation. So I told you that I will consider translation also because translation is very confusing to some people. So translation, before starting translation in my previous class, I had discussed with you the structure of transfer RNA. Okay. So the transfer RNA structure I had drawn, and then I had explained to you that transfer RNA is the molecule which brings the desired amino acid to the ribosome. Okay. So you can see in this diagram here, the transfer RNA is attaching itself to the ribosome, and it has an anticodon thing, UAC. Somebody was asking me, sir, why are you always picking same codon? I can pick any codon, right? Here I have taken methionine codon because it is the first one. So UAC for methionine. The codon for methionine is AUG, but for AUG you have an anticodon UAC. So UAC carrying codon, UAC anticodon carrying transfer RNA can only bind with the methionine amino acid. This methionine amino acid gets charged after reacting with ATP, and now it can form bonds with the ribosome. Alright. So this transfer RNA is bringing the correct amino acid to the ribosome, and it will bind itself to the it will form bonds, the anticodon will form bonds with the codon. So AUG, you can see AUG is the information present on the messenger RNA. See in this diagram carefully. AUG is the first codon for any protein. Methionine is the first amino acid for any kind of protein in the world, either whether it be bacteria, whether it be some mycoplasma, some other animal, lower animal, sharks, fishes, anything. All proteins have the first amino acid as methionine. So I'm beginning the transcription and the translation process. So after transcription comes the translation. So the first codon has to be AUG, otherwise it will not start. So AUG has an anticodon UAC and methionine is hanging upwards. So where will this methionine go? Methionine will lock itself in this, inside this amino acid binding site. You can see there are two sites here. In each of these two holes, these large red holes, in each of these two holes, one amino acid can sit safely inside. It's a place for accommodating the amino acids. So now first amino acid has been formed. After that, the second amino acid will be welcomed into this chain. Suppose the second amino acid is valine. So this is a catalytic site, and it will allow the formation of peptide bond. It will encourage the two amino acids to come together and form a peptide bond. When they have formed a peptide bond, this chain will move forward. This chain will move upward. Methionine comes out of the ribosome. Now valine will only be able to come out when it forms the second peptide bond with another amino acid. Okay, so it can only form a peptide bond when it receives another amino acid, right? So in this way, methionine, valine, suppose phenylalanine comes, after that amino acid like tyrosine comes. So in this way, the chain will grow in a further manner. Okay, so this is how translation is happening. Methionine, valine, histidine. So these are the abbreviations for the various amino acids, right? And in this way, this will continue until and unless the messenger RNA comes to the stop codon. As soon as stop codon comes, the ribosome will stop reading the genetic code, and it will say that the translation process is over. So the whatever the chain is there, the big chain of proteins containing several amino acids, it will be terminated exactly at that time. The chain will detach itself, the amino acid polypeptide chain will detach itself, and the translation process or the protein synthesis process is said to be complete now. Okay. So up till this part we have done in the last class. I am giving you the summary again. Complementary DNA strand and the template DNA strand. The template DNA strand makes the messenger RNA. The messenger RNA meets transfer RNA. Transfer RNA will bring the amino acids, and that will form the protein inside the ribosome, right? So this is the summary part. Okay. Regarding mutations also, this is a very short presentation on mutation. What is a mutation? Mutation is a mistake in the genetic code or the DNA code. So it results in a broken non-functional protein, and mutation can affect some cells also, and until and unless it has a huge kind of effect like a cancerous effect, the mutation is not normally harmful. But most mutations can be harmful because the radiations have an effect on some genes which cause them to become cancerous cells. Anyway, mutation can be of several types. Point mutation, base substitution. So base substitution results in the exchange of one amino acid for the other amino acid. Insertion amino acid, insertion means the whole genetic code, because it is commaless, punctuationless, whole genetic code will be changed. Deletion will also cause the whole genetic code to be changed. Okay. So suppose this is a funny thing that everybody can understand, right? So just now I have taught you what is mutation. So if point mutation is there, suppose the original word is the fat cat sat. Okay, this is making some sense, right? So how many people of you love cats? Raise your hands. How many people love cats? Some people are some people love cats, some people love dogs. I think I love both. I had a dog. My mom had a dog, and he died some years later. Some years from now, he died, and from that point, I haven't been able to take a dog anymore. I love feeding the street dogs and so much. I also offer milk to some stray cats and dogs also, but I haven't been able to do that. So how many people of you love cats? Just a casual question to you. Yeah, okay, so I think more centers have joined. Yes, the centers which have joined just now, please also report your attendance. Okay. Okay, Shillongs, I think, yeah, the students have increased now. Some people just came in. Girls, I believe, yeah, girls have come in. Okay, so centers, now you can report your attendance at present now. Yeah, Shillong, St. Paul's, Itanagar, and the other centers, please report your attendance. Right. Okay, so meanwhile, okay, I'm just asking you a question. How many people of you love cats? Nobody? How many people love dogs then? Dogs are more faithful, right? Cats, they can be, they cannot be. It depends. Cats have a very different kind of personality. But dogs always are faithful by rule. They never betray us, right? So how many people love dogs? Just raise your hands, or yeah, you can just reply on the chat now. Because chat is working fine now. Okay, yeah. Same as for me, also Itanagar. I love both. Okay. Anybody else? Okay. Okay. Maybe people are not trying to answer. Alright. Let's come back to the topic if you want me to continue with the subject only. So here is a sentence. The fat cat sat. The fat cat sat. Okay. It is making some sense. If there is a point mutation that, suppose I am replacing, I am deleting the E word. I am deleting the E alphabet, right? Or I am changing, sorry. If I am changing the point mutation, I am changing the substituting E by A, right? So if I am substituting E by A, due to point mutation, that becomes the fat cat sat. THA is a silly word. It does not make any sense. But the other things do make sense. If it is insertion mutation, okay. So suppose I am inserting the letter double T, then it becomes the FTT, ATC, ATS, AT. It does not make any sense, right? So these things happen. Deletion, if I'm suppose deleting the letter A in fat word, then it becomes the FTC, ATS, AT. So it does not make any sense. So mutation causes large amount of changes. Point mutation might have very few effects, but insertion mutation and deletion mutation, they can change the meaning of the protein chain altogether. The protein will be totally non-functional now, because a totally different protein will be formed now. Okay. This is just to educate about you for the mutation part. So there are some mutations, okay, like CTC changed into GAG, and there is no effect on protein. And mutation 1, glutamic acid change, there is no change. Mutation 3, changed by Valen. Okay, so hemoglobin mutation, you know it's called sickle cell anemia. So sickle cell anemia happens due to change from CAC into GUG, and it causes a very large amount of change. The protein is not any more soluble in water, and it causes sickle cell anemia. Okay. So this is about what I wanted to show you about the various aspects of translation and transcription. I have one more slide show that I thought you might be interested in. So this is another I prepared for some students. So DNA replication, I am including DNA replication in this also. DNA replication happens by the help of helicase unwindase enzyme, just for revision. And DNA replication is happening in this way. Okay. And after that, transcription and translation, central dogma I taught you in this chapter. RNA I told you already about. RNA has uracil instead of thymine. 3 types of RNA: messenger RNA, ribosomal RNA, transfer RNA. All this I have already told you very well. And messenger RNA, it is having this structure. Ribosomal RNA is having this structure, and transfer RNA has this form. Okay. So this PowerPoint presentation, do you want this? Okay. I believe it will be helpful for you. And now today we will basically discuss the translation and the splicing part is also discussed in this slide, that what is splicing. We are removing introns. So for those students who don't have the notes or want the images also, this will be very helpful for you. Okay, junk sequences, splicing, everything is explained very well in this diagram, and this is a short summary kind of notes. Do not think that they will be as effective as the notes I have given you, but this will be very useful for you for learning things in a very quick manner. So within 5 minutes you can easily revise the chapter. Okay, so this I have already told you in the beginning of the class. Okay, so this is about transcription, translation, and now today we have to begin with the Lac-Operon model. Okay, the Lac-Operon model. Okay, so now put the heading, the regulation of gene expression. The regulation of gene expression. We have studied how DNA is making RNA and RNA is making protein. But what we have not discussed is how the protein regulation or the different genes, they are being regulated to make some things and not to make some things. Because up till now what we have done is making things only. But we also, when we know how to make a thing, we must also know how to control that ability. Okay, because we do not want to produce anything in excessive quantities. We want to make it in a controlled manner, that we have to make resources only when they are needed. Otherwise, it will be a wastage of resources. And cell does not waste any resources. Okay. So please now put the heading, regulation of gene expression. Regulation of gene expression. Regulation of gene expression. Okay, so please write now. Regulation means the ability to control. Regulation refers to the ability to control the expression of genes. The ability to control the expression of genes. Okay. So regulation can be done at many steps. Regulation can be done at many levels. Such as where are the points then where the regulation can be controlled. The first point is at transcription level. At transcriptional level. Okay. So for transcriptional level means making making RNA out of DNA. So formation of primary transcript. Formation of primary transcript. That is while making hnRNA. Okay. Second is the processing level. Processing level. So what is the processing level? Processing level means regulation of splicing. Then we are going to cut the introns, right? Regulation of splicing. Regulation of splicing. Splicing, everybody remembers? Splicing, everybody remembers? Just a second, I'll show you again in case you forgot what is transcription. Okay, this is splicing. In case you have forgotten, this is splicing. Capping, tailing, splicing, all these three things have to be done at the same time almost. Okay, so after capping and tailing, we remove the splicing, we remove the introns, and this process is called splicing, and then we get only the coding sequences. And this finished ribosomal RNA, it is called the messenger RNA, and only messenger RNA with the cap and the tail, it will be able to come out of the nuclear pore. So I hope you remember this. So this is about splicing. Okay. The third level is at the level of transport of messenger RNA, at the transport of messenger RNA from nucleus to cytoplasm. From nucleus to cytoplasm. Okay. And the fourth level is the translational level. Translational level. Okay. So these are the four levels where I can stop the or increase the or decrease the level of formation of messenger RNA or make the protein. These are the four levels where I can control the formation of a protein, and this is how control is exerted. Right? Now let's come on to the next part, how the cells are controlling translation. Okay. So please finish it quickly and then we'll move on further. Please write it down quickly. Okay. All right, so I believe you have finished it. Okay, so let's start what is happening now. Please write now. The genes in a cell are expressed to perform a particular function, or a group of functions, Okay. So if a cell is producing something, okay, it is only made possible by the help of genes. Okay, so the genes will decide what thing to make and what not to make, right? So the genes will only be able to decide this thing. Okay. So what we are going to consider now today is the particular case of beta galactosidase gene enzyme, sorry, which is able to make lactose, okay, and able to break down lactose. So we'll study about this gene today. For example, if beta galactosidase enzyme, if beta galactosidase enzyme is synthesized by E. coli, it is used for breaking down lactose. Lactose is a disaccharide made of glucose and galactose sugar. Okay. Breaking down lactose into galactose and glucose. Okay. Because both galactose and glucose, they are energy rich substances. Both have the molecular formula of C6H12O6. Galactose is an isomer of glucose. Okay. Both are monosaccharides, and lactose is making these monosaccharides, so it is called a disaccharide. But and used as source of energy, by bacteria. Right? But if lactose is not present, then we do not need to make beta galactosidase enzyme. Okay? But if lactose is absent in medium, then bacteria will turn on to a different energy source. Maybe it will break down some proteins, maybe it will break down some starch. Okay? But if lactose is absent in surrounding media, medium, then there is no requirement of making, because it will be a waste of enzyme, making beta galactosidase. There is no requirement of making beta galactosidase in that case. Right? So what will we do now? Okay. This is a very simple this condition of having or not having having or not having lactose, this condition of having or not having lactose is the fundamental basis of regulation of gene. Basis of regulation of gene. What does this mean? That if I am having lactose, then I need to make the mechanism for making beta galactosidase. But if I do not have galactose, I should not make that enzyme. Understand? So what I'm trying to say here is that making or not making, okay, making or not making something is a function of, is dependent upon the environmental conditions. Okay, so if the environment allows it, I will make it. If the environment does not allow it, why will I make it? It will be a wastage of resources. So the gene expression, the gene regulation is based upon this funda, right? So now you can deal with one example of gene expression, and we will do it in bacteria first. Please write now, next point. In prokaryotes, control of rate of transcriptional initiation is the predominant site for control of gene expression. Site for control of gene expression. Right? What I'm trying to say is, if I can control the basis of enzyme formation, like, I will scroll up. I believe you have it not written. So if I can control the rate of transcriptional initiation, that transcription meaning making messenger RNA. So if I can control when I'm going to make the messenger RNA, I can also control the enzyme that is going to be formed. Okay, this is the basic thing, right? So now my function is to study what is a transcriptional unit in the basic gene. So please write the next point. What is a transcription unit? I have to study now, because we are studying transcription now. The transcriptional control now we are studying. Please write, in a transcription unit, that is the part where DNA is making RNA, transcription unit, the activity of RNA polymerase is regulated by accessory proteins. Okay. There are some other proteins which are able to control this phenomenon, accessory proteins. Okay. Which are affecting the ability to start to recognize start sites. The ability to recognize start sites. Right? Now there can be a positive control also. There can be a negative control also. Please write this quickly. Please write now. The regulatory proteins, next point. The regulatory proteins. The regulatory proteins can act positively, activators, and negatively, in the form of repressors. So it can be activator, it can be repressor. Now we will study this in the following section now. Okay. Next point. The accessibility. This is an important point. The accessibility means the ability to reach. The accessibility of promoter region of prokaryotic DNA Now you might ask, sir, why are you always writing prokaryotic? Why are you not writing eukaryotic? The reason is because most of this research is being done only in prokaryotes. Because eukaryotic transcription and translation is very, very difficult to study. We know the basic fundamental framework, but we do not know the exact steps in which it is happening. We do not know the exact phenomena, the stepwise phenomena. In prokaryotes, it is comparatively easier. In eukaryotes, it is very hard to study, because eukaryotic protein synthesis is also very slow. Okay. So the accessibility of promoter regions of DNA, promoter regions of prokaryotic DNA is regulated by interaction of proteins with specific sequences called operators. Regulated by interaction of proteins with sequences called operators. So this will be asked many times in your board examination. What is an operator? Okay. Now next point. I'm going to explain you the position of operator with respect to promoter and the structural gene. So please write. The operator region is situated adjacent to the promoter elements in most operon, in most operons, okay, the operator and promoter are situated very close to each other. So I'll just give you a diagram also. I have a, yeah, just a second. I have a PPT for you also for regarding this. Okay. So, just a second. So, this is the operator side, okay, and this is the promoter. This is the promoter, and it is labeled by, look at the position here. O is the operator, and the promoter is just located adjacent to it. Okay, how this is going to work, we are going to study in this manner. Okay, so but just just a second, just please try to look at the position. Operator, promoter, and after that is the ZYA sequence. That is a structural gene. I'll come up to that later. Okay, I'm just trying to show you what is present here. Okay. All right. So it is located adjacent to the promoter in most cases. So please now write the next point. Next point. In most operons, and in most cases, the sequences of the operator binder repressor protein, binder repressor protein, binder repressor protein. All right, binder repressor protein. Each operon has its own specific operator, and own specific repressor. I will explain to you in detail how these things are working. So I have all the cases. I'll just show you in the diagram, because if I draw the diagram, it will take a lot of time. So I have made a PPT, and it will be very easy for you to learn that way. Okay, so don't worry about it. This all things will come easily. Don't worry. Lac operon is a very difficult topic to understand, and I'll make sure that you understand it very well. Okay, don't worry about it. Now, please write an example. For example, lac operator, I'll choose a different color for it. Lac operator is present only in lac operon, is present only in lac operon okay, and it interacts specifically with the lac repressor only. Lac repressor only. So please finish this quickly and then we'll begin the final explanation part, how these things are working together. So please finish it. Finished, right? Okay. I want to end this chapter today. Hopefully I can do it. Okay, this is some message. All right. So let's try to understand how this is all working together. Okay. So the lac operon model, why do we study lac operon or any other model for that purpose? The reason is very clear. We want to understand how the genes are being controlled. Because if we try to understand one gene, it will give an idea of how the other genes are being controlled in our body or any other organism's body and so on. So, for example, each cell in the human genome contains all the genetic material for the growth and development of a human being. Okay. For example, your skin cell, if I take that out and I try to make a clone of you, okay. So suppose you have a clone of you. So what will you do with your clone? Okay. Even your skin cell is sufficient to make a clone of you. So can you, until now human cloning has not been achieved. So if it were possible, so we can think of clones doing our work, clones going to college for us, clones working for us. So maybe it will be like a slave, maybe it will be like a friend to us. So because we understand ourselves better than anybody, right? So maybe we can make it a friend and ask our friends to work for us. Okay, so some people who watch Naruto, they might remember the Bunshin Jutsu of so many anime characters, right? So they make a lot of clones and they make them do some work or something like that. Okay, risky jobs also done by that clone also. So the thing is, if I take any cell, it contains the complete genetic information of all the things that are happening in your body, right? So, but not every gene has the ability to express itself. For example, your skin cell, it has the same genome that is present in your pancreatic cell, which makes insulin. But your skin cell is not making insulin. Skin cell is not making insulin. Those genes are turned silent. We do not want them to express itself. Okay. Why? Can anybody tell me why? Why do we not want the skin cell to make insulin? If it were happening, we would not have the need to take insulin from outside. If somebody has diabetes, if our skin cell was also making insulin, then we will not need to take insulin from outside. So why is this condition existing in our body? That some things are making different things. So every cell is making a different function, or tissues are doing different jobs. So why are they doing this like in this manner? Why isn't a skin cell making insulin, or a hair follicle cell? Why does it not make melanin and at the same time does not make insulin? Give it a guess. If you're wrong, I'll correct you. Why doesn't a skin cell make insulin? You can give general answers also. I don't need very specific answers for this question. Students, please respond. No idea. Okay, nobody has any idea about this thing. I'm asking you, look, it is written in the slide also, that each cell in the human body contains all the genetic material for growth and development of a human being. Just like if I take a skin cell, if I analyze the DNA, it has the same DNA as any other cell in my body. Okay, but some genes are silent. And okay, somebody is saying it will not reach the target site. No idea. Okay. Thing is, try to understand the basic hierarchy of animal kingdom or the plant kingdom, or the basic hierarchy of biological classification. Prokaryotic cell, eukaryotic cell, single eukaryotic cell, protista, multicellular eukaryotic cell, fungi. Then tissues are forming plants and animals, tissues forming organs, organs forming organ systems. This is the basic organization, right? So when we make tissues, when we make organs, what is the thing that happens in that organism's body, like hydra, sponge, or let's take a higher organism like ascaris or tapeworm. Thing is, tissues are dividing their functions. I'm asking a skin cell for protection only. I'm asking a teeth cell for hard cells only. I'm asking a bone cell for hard tissue only. I'm asking a muscle cell for making contractile protein only. I'm asking a hair follicle cell to make melanin and keratin only. Melanin for coloring the hair, keratin for making the substance of hair. So each cell has its own proper function. But if you are forcing every cell to go independent and make everything by itself, it will be a huge wastage of resources. The organism will also die, and there is no point in making everything. If you make excess of insulin, the body will go into insulin shock, glucose shock. The body glucose level will suddenly drop down. Insulin is also not taken in excess quantity. If you take it in excess quantity, a person will die on spot. If you given a healthy person some insulin, he might die on spot. That is the thing. So insulin basically, or any other cell, basically does not make everything. Because if it does, division of labor will be lost. Okay. If you're not still able to understand this, try to imagine this. If you go to your home right after this class now, okay, when you go back to your home after your class, and suppose your mother says, please cook the food of each and everybody. Okay. Or your mother says, you should do your own food, you should cook your own food and wash your own clothes, clean your room and everything. So every person has to do everything. Your dad has to do everything also, your mother has to do everything also, your younger sisters and brothers will do everything also. So is this going to increase the efficiency? Yes or no? Is this arrangement, suppose you're going home and your mother asks you to make some food. So is this arrangement going to help you? No idea? Or do you get an idea what I'm trying to say here? Everybody in the house has some particular function, has some particular work. For example, we are always asked to make some small things and fill the water bottles, for example, buy some vegetables from the grocery market. Okay. So if you are asked to cook food also, buy the groceries also, and your mother is also doing the same thing, she is earning also, she is working also, and she is doing everything at home. Everybody has to do their own work. That will defeat the purpose of being in a home. If you're going to your home and everybody has eaten except you, and you're asked to make your own food, will you revolt or not? Come on. Is it such a difficult question? Do you understand my point? Division of labor is done with a purpose. That each functional unit in a house or in a city has its own specific function. They're specialized for that function. But if somebody is asking you to do everything, then that is going to decrease the overall efficiency of that unit. Right? It's just like amoeba. Amoeba has to do everything by itself. But if it has living with other people, they will normally, for better efficiency, some cells will do some things that are adapted to their function and purpose. Other cells will also do that, will also have a specific function. The functions will not clash with each other. They are tended to support each other. For example, epithelium cell gets its oxygen with the help of blood cell only. It cannot do that by itself. Although it's close to the air, although epithelium cells are close to the air, they do not exchange oxygen. Do you breathe through your skin? Do you breathe through your skin? I'm asking you. Yes or no? Can you breathe through your skin? Maybe for a small amount. Can you? Don't say no idea. Give me an answer. Yes or no? Yeah, we cannot do that. Only frog can do that with a moist skin. Okay. So our skin does not have this function. Our skin does not have this function of performing the respiration. It is a function of the lungs and the supportive blood which carries the oxygen and to all the other parts. Okay. So we have a circulatory and respiratory system for that. Okay. So this arrangement is going to increase the efficiency. Okay. Cells working and coordinating with each other. That is what forms an organ. Tissue is a group of different cells, many cells performing the same function. And organ is a group of tissues, and many organs form organ systems. So this is the basic hierarchy. Cells performing better in a better manner by outsourcing their other needs to other cells. Okay, they will do their function properly, and they will ask the other cells to function properly also. That is how a multicellular organism body works. Your heart does not say to your muscles that you are always having some rest and I have to work for 24 hours a day. Heart works for 24 hours a day for the period of your life, for whatever years you are there. But we do not use a muscle so much, hand muscles or leg muscles so much. We cannot do that. So each and every cell has its own specific function. We cannot put the function of heart in the muscles. Okay, we cannot do that. So that is the basic thing you should understand. Some genes need to be controlled. They need to be expressed all the time, and most genes must be suppressed. They do not need to function all the time. That is the basic thing. Right? Okay, so the control of gene expression, some genes are not expressed and they are switched on and off required according to their specific need. Last point, please read it again. They are switched on and switched off according to the requirement, according to the need. Now let's come on to the operon. Okay, so operons were discovered by Jacob, Monod and Loff. Jacob and Monod gave the lac operon model. So these people are given in this picture here. They discovered Jacob and Monod discovered the lac operon model. So what is an operon? Operon is a group of genes that is transcribed at the same time. I'm giving you these notes. I'm giving you these PPTs. Don't worry about that. Try to understand this definition. Some genes are transcribed. They are made at the same time. And they normally control an important biochemical process. Operons found only in prokaryotes. We do not have full understanding of eukaryotic genes, right? We do not have full understanding of eukaryotic genes. So up till now, we will discuss about operons in prokaryotes only. First discovered or well understood example of operon was lac operon. First well understood example of gene expression was seen in lac operon in E. coli. So it consists of 3 genes, and each of the 3 genes are involved in breaking down lactose into glucose and galactose. So one of these things is the gene for enzyme called beta galactosidase, right? And this enzyme is able to hydrolyze lactose into its components, right? Okay. So I'll give you this PowerPoint presentation. Don't worry about that. You will receive it by today. Okay. All right. So adapting to the environment. Try to read these things. E. coli can either use glucose, okay, it can use lactose also. But the thing is, to process lactose, we need to break it down because it is not a simple molecule, it is a semi-complex molecule. So the bacteria wants glucose, but glucose is not available from lactose readily. So first you have to break it down. So 4 situations are possible. So we don't use cell phone, it's difficult for us to connect with the information. Okay. Alright, Itanagar, I understand your problem. Okay, I'll do it slowly, that you are able to write it. Okay. If you're not using a cell phone, yeah, I understand your problem. So you can basically take this information from me in the class then. So for those students who are not using cell phone, I'll do it a bit slowly. Okay. So lacopron, you can write these information down if you are not using a cell phone. It's okay. It's perfectly fine. It's okay. Okay. All right. So Tetso College has just joined us. Okay. So what I'm trying to say here is lacopron has 3 genes. One of these genes is beta-galactosidase that we were just talking about. Okay. So E. coli can use glucose also, lactose also. But lactose is has to be broken down into glucose and galactose. Then the bacteria can use the lactose. Otherwise, it has no use. Otherwise, it cannot use lactose sugar. So we need to digest the lactose. How will we digest it and what will we make out of it? We will make it by using an enzyme called beta-galactosidase. So please write it down quickly and then we will move on. Okay. All right. So next slide, please see this. 4 situations can be possible. If glucose is present, if anybody is left out and you're not able to copy it, I think somebody is left out. Okay. Yeah. Please do it quickly. So, okay, I hope you have done it. Okay, some people are still writing, so I'm just waiting for you to finish writing the slide. Try to write it fast. Okay, now you have finished it, right? All right. So now there are 4 situations that are possible. Please keep writing. If glucose is present and lactose is absent, then in that condition, we do not need to digest the lactose. If glucose is present and lactose is absent, I do not need to make beta-galactosidase. There is no requirement. Okay. So in this condition, we will not make any beta-galactosidase. No requirement. Wastage of resources. Second condition. If glucose is present and lactose is also present, then what is my preference? I will still prefer to use glucose, okay, because already glucose is present. Then there is no need to waste resources on a difficult substrate. Why do we need to digest? Suppose there are two things available to you, and both of them have the same purpose, providing energy. So you will prefer the thing that is easily digested, right? So that is the same case with bacteria. If glucose is present and lactose is present, it will not break down lactose. It will only absorb the glucose. Third condition, glucose is absent and lactose is absent. In that condition, it will also not produce beta-galactosidase. These are the negative conditions. In these conditions, all these three conditions, beta-galactosidase will not be produced. Okay? But then there is the fourth condition. This is the fourth condition. If glucose is absent and lactose is present, in that condition only, E. coli decides to make beta-galactosidase. In the fourth condition only, we will remove the controls for not making the enzyme, and this is what we are going to study in the next part. Okay. So please tell me as soon as you have finished, Itanagar and Shillong students who are writing this information. Please tell me as soon as you have finished. Okay. As soon as you finish, please let me know. Can I move on to the next slide? Okay. So now we must study the control of lacopron. When lactose is absent, the first case, no beta-galactosidase. Okay. So please see this figure. In the DNA has a regulator gene. Okay. It is making us something called repressor protein. Repressor protein is something which is being made continuously. I am continuously going to make some repressor protein. Alright. And the repressor protein is blocking making of blocking the entry point of this blue colored heart shaped structure. This blue colored heart shaped structure is the RNA polymerase enzyme. RNA polymerase enzyme wants to attach itself to the operator site. Like the operator site is the entry point. Without attaching itself to the operator, RNA polymerase cannot make any enzyme. RNA polymerase cannot make any RNA. So that is blocked by repressor protein. So this is the first case. A repressor protein is continuously synthesized. It sits on a sequence of DNA just in front of the lacopron called the operator site. Please, yeah. So please write this down quickly. And you have to draw the diagram also. Okay, you have done it. Let's move on to the next slide. Next point. The repressor protein is blocking the promoter site where the RNA polymerase settles before it starts the transcription. The repressor protein, it is blocking the, write the next point also. Repressor protein blocks the promoter site, okay, where the RNA polymerase settles before it starts transcribing. Look, between O and Z, there is a promoter. I'll just show you in the next figure also. Promoter is not labeled here. It will come in the next slide. Okay. So now let's move on to the next slide. Repressor protein blocks the promoter, right? Now when lactose is present and glucose is absent, a small amount of sugar called allolactose, it is formed within the bacterial cell. A small amount of sugar called allolactose is formed within the bacterial cell. Okay. These figures are serving as your notes also. Okay, so everybody you can who are writing the notes, they can please write these statement also, because I can see some people are not writing. Okay, so I understand you want to take the PPT slide from me, but if it's possible, try to make some notes out of it. Yeah, I'll already give you this, no problem with that. So when lactose is present, there is a sugar called lactose or allolactose. It is formed within the bacterial cell and it attaches itself to this brown colored compound called repressor protein, which is blocking the yellow colored operator site. So this allolactose causes the protein to change the shape, and if the shape of protein changes, in that condition it cannot attach, it cannot block the operator anymore. It will get free from the operator site. Okay, protein to change in shape, it's called conformational change, and now it can no longer sit on the operator site. And because of this, read the second part, RNA polymerase can now reach the promoter site. In the previous slide we saw RNA polymerase was blocked, right? In this next slide we can see here, now the RNA polymerase can reach its target site. Okay, done? Can you explain this part again? Look, please try to compare this diagram with the previous one. In the previous slide we can see repressor protein is blocking the operator. Okay. Because of this problem, because of this roadblock, yeah, everybody has seen in the Northeast has seen roadblocks, right? So some people they try to block the road that we will not allow you people to enter. Like for example, the government authorities they face a roadblock, so they cannot reach the place where they want to be. They cannot redistribute the rations, the daily supply goods and other things to the people, right? So suppose the repressor protein is acting as a terrorist. It is acting as a roadblock. It is preventing the RNA polymerase from reaching its target site. Okay. Now see the next slide. This double hexagon, okay, it's a double benzene-like structure, and this double benzene-like structure, it is called allolactose. Allolactose will bind itself to this repressor, brown-colored repressor protein, and when these two bind together, there is a change which happens in allolactose. Sorry, in a repressor protein. The repressor protein is having this structure initially, and after binding with the allolactose, it changes its structure in a different manner. It changes its structure, and when it changes its structure, it cannot bind to the operator anymore. If allolactose is removed, then it will regain the structure, but until that time it will change the structure and it cannot bind to operator anymore in this condition, right? Let's see see this diagram now. You are seeing what is happening, right? Allolactose is binding with the repressor. In the next condition, what is happening? The repressor protein leaves the operator. New diagram now. The repressor protein is leaving the operator site. Now the blue colored compound called RNA polymerase, it can now attach to the promoter. Okay, I hope you understand it now. Right? So now RNA polymerase needs to reach promoter. But before it can reach the promoter, it has to pass through operator. So operator is a gateway, right? Yeah, I hope this diagram helps you in understanding. I made these diagrams so that you can understand this. Otherwise, if you read the NCERT, it is very confusing. So I made this part so that you can understand this concept in a better manner. So you can draw these diagrams and these will help you in understanding what is happening. Okay. Yeah, Shillong students, please settle down quickly. Yeah, I can see you are moving. Okay. Alright, so I hope you have drawn this diagram now. I'll move on to the next slide. Now when both glucose and lactose will be present, in that condition we will shut down the lactose mechanism. Okay. So suppose, just a second. When glucose and lactose are present, RNA polymerase can sit on the promoter side, but it is unstable and it keeps on falling off. Okay, that can also happen if both glucose and lactose are present. In that condition, RNA polymerase keeps on falling, keeps on coming back. But if glucose is totally removed, then in that condition, the cell has to depend on lactose only. You can write this also. It keeps on falling off, it keeps on sitting again. So please draw this quickly. Okay. Let me know as soon as you have finished the diagram. I hope you have done it. Okay. Moving on. Shall I move on? Okay, no protests so far. Okay. Now, if glucose is absent and lactose is present, in this condition, you can see what is happening now. Activator protein helps the RNA polymerase, and there is straight away transcription. The RNA polymerase can move on to the operator site, and now it can bind to the promoter. That RNA polymerase has to first pass the operator, then only it can reach the promoter site. Promoter is the actual target. Operator is just the roadblock. It has to pass in any case. So if the roadblock is cleared, in that condition the RNA polymerase will be able to reach the promoter site. So I told you the 1, 2, 3, 4 cases. This is the fourth case, and in this fourth case you can see what is happening. There is a very smooth movement of RNA polymerase over the structural genes. No obstruction at all. No obstruction at all. It will very smoothly move towards the transcription site. It will make 3 products: beta galactosidase, transacetylase, and permease. So I'll explain this in the next part. Another protein is needed called an activator protein. This protein stabilizes the RNA polymerase. It only works when glucose is absent, and in this manner E. coli bacteria makes enzymes to metabolize other sugars in the absence of glucose. Okay. So please do this part quickly. All right. So Yeah, the theory part is good. You just need to make the diagram. Theory part is easy. It's 3 lines you have to write. The diagram you have to draw carefully. Please show the transcription. It is very important. So if somebody asks you about Lac-Operon model, you will draw the first case and the second in the fourth case only. That in absence of that in presence of glucose, lactose metabolism is not required. If lactose is absent, the gene is shut down, switched off by binding to repressor protein. But if the medium has lactose and glucose is absent, in that case repressor will be removed. Lactose or allolactose, both the sugars can bind to repressor protein and can remove it from operator site. In that condition, RNA polymerase is free to start the transcription. Okay. So I hope you have done it. Okay. Yeah, I can see some people are, yeah, Shillong students are still drawing, I believe. And St. Paul's, I believe you are also drawing. Okay. Now I believe most people have finished it. Shall I move on? Shillong students, you have done it right? Okay, St. Paul's, you have also finished? All right. So this is a brief summary. Glucose is present, lactose is present. Activator is absent. Repressor protein is lifted off, no transcription. Okay. Glucose is present, lactose is absent, no transcription. Glucose is absent and lactose is also absent, no transcription. But if glucose is absent and lactose is present, in that condition, activator will be bound to DNA, repressor will be lifted off the operator site, and in that condition, RNA polymerase is able to undergo the transcription. Okay. The fourth case, yeah, you can draw this chart. This will help you very much in understanding the conditions. I have already explained this. This is just a summary. Okay. So now you will understand what is operator, what is promoter. I hope you can write these definitions down also. Okay. Now I need to make one more diagram after this. That I will do in the notepad. So after you finish it, I will continue. So in the previous diagram that you have just drawn, you just have to make some change, make some additions. That is the only thing you have to do. So yeah, please finish it. The summary chart is not very hard to make. All right. So yeah, so please let me know as soon as you finished. You need to make 5 columns into 5 into 5 columns, 5 into 5 chart. And then I'll make another diagram, and then this Lac-Operon is finished. We'll try to finish this chapter today. So please hurry up. Okay. I'll repeat, glucose, lactose both present, activator not bound to DNA, repressor falls and keeps on falling off, and RNA polymerase also falls off, no transcription. Okay, glucose present, no transcription, glucose, lactose both absent, no transcription, but in this condition, activator will be bound to DNA. Only in absence of lactose and only in presence of lactose and absence of glucose, activator can bind to DNA. Okay, or in just simple way, if glucose is absent, only in that condition activator will bind to DNA, and if activator cannot bind, the RNA polymerase will keep on falling off. So it needs a stabilizer. So activator acts as a stabilizer. Without stabilizer, it cannot continue its function. Finished St. Paul's and Itanagar Tetso College. Okay. I think everybody has done it now. Now there is a small difference in your NCERT and the diagram I have drawn just now. The diagram, there is a small thing. In the NCERT, they have done some more things that I have not, like the diagram was, I was making this diagram but could not complete it because the space was becoming too narrow and I wanted the space to look wide away. So in this diagram here, I am going to just make some changes now. So you can just, this diagram is, in this condition, in this diagram, you can replace by another diagram that I am going to draw. Okay. This diagram, this one, you can replace by the diagram I am just going to draw now. Okay. So, yeah. Now I need to give you this diagram now, the full diagram. We have to draw a long sequence of messenger RNA. Okay. P is the promoter, initial promoter of another gene or the same gene, inducer or repressor. This is the promoter site and this is the operator site. Okay. This is the operator site. Let us label it O. And what is happening here is that inducer is making a specific molecule. It is called repressor messenger RNA. Repressor messenger RNA. In NCERT, activator protein is not mentioned. In NCERT, activator protein is not mentioned. So that was perfectly fine if you don't want to write about it, so perfectly fine. Repressor protein makes repressor messenger RNA makes repressor protein. This repressor protein attaches to the operator site. Attaches itself to the operator site. Under this condition, the RNA polymerase cannot bind to the promoter site. Okay, so you can write here, a repressor binds to operator Repressor protein binds to operator and prevents RNA polymerase. You have already done it, just repeating it. This is just given you NCERT, just trying to explain it. Prevents RNA polymerase. This diagram is what you will draw if it is being asked in your board exam. This diagram is what you are going to draw if it is being asked in your board exam. All the other diagrams I have given you just for explanation. Otherwise, you would not be able to understand this diagram. Prevents RNA polymerase from transcribing operon. Transcribing operon. This happens in absence of lactose or allolactose. In absence of lactose sugar or allolactose, this is going to happen. RNA polymerase cannot bind, the same structure that we were drawing initially. This is RNA polymerase. Okay, it cannot bind to the promoter operator site in absence of lactose, allolactose. Lactose and allolactose are together called inducer. It is called inducer. In absence of inducer, RNA polymerase cannot bind to the promoter site. Now please draw 3 more genes, Z, Y and A. Alright, Z, Y and A. It's very easy, nothing special in it, it's really easy. Now, this is the initial condition. Now the next condition is happening. This is the initial condition. Second condition, let's label it A and B. This is condition A. Now the second condition B, in presence of inducer. In presence of lactose or allolactose, called inducer. And everybody remembers how in the previous diagram we drew lactose and allolactose like this. Just a second. Yeah. So double hexagon like structure we had drawn previously. This is allolactose, or in absence of inducer we need not draw, but now in the presence we need to draw it. Okay. So now in the presence of lactose or allolactose, the repressor molecule, it will bind to the inducer. So now for the second step, the B step. P, I, P, O. Okay. Now the operator block has been lifted. Now the RNA polymerase can bind itself to the promoter site. Why is this happening? Because repressor protein has changed its structure. The repressor protein, it was lens shaped, now it has become triangular. It cannot bind itself to the promoter. It has become bonded with this thing. This repressor molecule, it has bound itself to the lactose molecule. Now it cannot bind itself to the promoter. Now it cannot bind itself to the promoter. So until and unless lactose exists in the medium, RNA polymerase cannot bind. Until and unless lactose exists in the medium, repressor cannot attach itself to the operator site. Now RNA polymerase will begin the transcription process. Transcription. It will happen now. And now the three structural genes, gene Z, gene Y, gene A, all these three genes, they will now make their messenger RNA. And this messenger RNA, it is like this. Now I told you what is an operon. Operon is a group of genes that are expressed together. So gene Z, gene Y, gene A, all these three will be expressed together at the same time. Now see this. You have to see this. Z, this is the lac messenger RNA. Lac messenger RNA for the lac operon. From lac operon genes. Okay? Made by these three group of genes. So this process is called transcription. Transcription is happening. Right? Now for the last part. Z, Y and A, they will show their effects. Finally, lac Z makes the most important enzyme called beta galactosidase. This enzyme is the one responsible for breaking down the sugar called lactose. Permease enzyme, this is another enzyme. It increases the permeability of lactose to the cells. If it is present, then lactose will be able to get inside faster. Third enzyme is not very well understood. People say that it has some role, but the role has not been discovered till date. We do not know exactly what this enzyme does. So it has a supportive role, but we do not know the exact function of this enzyme. So this step is called translation. Translation step. Translation of Z, Y and A. Okay. Label this also. Inducer, repressor complex. You can say this is the inactive repressor. Beta galactosidase, please write down. Beta galactosidase breaks down lactose into glucose and galactose. Permease enzyme, I hope you have done it. I don't think somebody is left out, right? If you want me to scroll up, let me know. Okay. Permease enzyme increases the permeability of lactose to the cells. Right? So these two enzymes are very well understood. So that is why I am giving you the details of this. Trans-acetylase plays a supportive role. Trans-acetylase enzyme plays supportive role. Okay. Now suppose by the action of these enzymes, all the lactose in the medium is finished. The E. coli has eaten up all the lactose in the medium. Okay. In that condition, the repressor will become active again and it will again block the enzymes. Last point of this lacopron, last important point. If lactose is finished by action of these enzymes, then inducer will separate from repressor. Repressor protein, and it again blocks the promoter site by attaching to operator. By attaching to operator site. Transcription and translation will stop. Transcription and translation of ZYA will stop. Finished. Lacopron model is now finished. If you have any doubts, please bring them on. We have very little time left now. Students, if you have any doubts, let me know. Okay. So do you have any doubts regarding this? This is the last thing. Short summary. All right. Short summary is this. If lactose is present in the medium and glucose is absent, then transcription will begin. Okay. I've already given you the summary many times. I will explain this again. What I'm trying to do this here, I'm trying to explain how and why we will begin gene regulation. So why are we regulating the gene? Because we need to make some things and we do not want to waste anything. If lactose is absent, why do we need to make enzyme for breaking it down? It will be wastage. So when lactose is absent, in that condition, repressor will block the operator site. If lactose is present, in that condition, inducer will block the repressor. It will change the structure of repressor and the repressor cannot bind to the operator site. Operator becomes free and RNA polymerase also becomes free. RNA polymerase will begin the transcription of ZYA. ZYA will form 3 enzymes. These 3 enzymes will help in breaking down lactose sugar. And when the whole lactose has been finished, suppose lactose finished after this, after the making of these enzymes, in that condition, I will again remove this repressor. In that condition, I will again remove this repressor, and in that condition, repressor will again bind to the lactose promoter and operator, and it will stop. So step A will repeat after that. If lactose is finished, then step A will come. If lactose comes again, step B will happen. This is the summary. Okay. Now please put the heading Human Genome Project. Okay. Human Genome Project. This is the last topic of this chapter. Human Genome Project. I hope everybody has been able to understand the Lac-Operon model now. It's not very difficult if you understand the diagram with the help of the figure. Do you still have any doubts? Please let me know. I will be able to help you out right now if you ask me now. Otherwise, I will move on with the Human Genome Project. Students, are you able to understand it? Yes or no? Lac-Operon. Lac-Operon is a very important topic. Please read it a few times. Please read the notes that I have given you a few times. You will be able to understand it. Okay. In NCERT, activator is not mentioned. Nothing else is missing. Okay. I have given you the full notes, more than what is required. And if anything is asked from molecular basis of inheritance in your exam, in your board exam or your NEET exam, nothing will come outside of my notes. That I can tell you. Okay. So please read this again and again, and you will be able to understand it better. So Human Genome Project, it will take some time. I think, yeah, my time is finished, so I won't waste your time right now. There are a lot of things that need to be explained in it. So I will bring up PPT for this also, Human Genome Project. Okay, and that way I think you are able to understand better. So I'll bring up PowerPoint presentation I'll make a PPT for this also. So in the next class, this chapter will be totally finished. Okay. So please solve the questions from your schedule, from your whatever assignments are being given to you. I will try to give you some questions as well. So please solve these questions. Please solve them again. And if you have any doubts, please reach out to me on the WhatsApp group, or reach out to me in whatever way you can, inside the class, outside the class. Okay. So if you don't have any mobile phone, please ask your friends to post any doubt, and I'll answer that in a short period of 1 or 2 hours whenever I am near my mobile phone. Okay. So thank you, dear students. Thank you for your time. In the next class, I'll start by Human Genome Project, and this chapter will be finished. Good night, everyone. Take care. Reach home safely. Thank you.